Good morning, and I hope everyone is having such a good time at the Festival of Genomics as I'm having. I'm Stephen Andrews. I'm a co-founder and chief scientific officer of Abigen, and I'm joined by Mansi Jain, who's a VP. And we're going to give you a little bit different talk today to hopefully answer some of the questions that commonly come up and really tell you why spit matters.
So Steve, you keep telling me spit matters, but I'm a researcher. I've used blood like most people do, and quite frankly, blood is a gold standard. You can extract everything you need from proteins, cells, DNA, RNA, everything is available in blood. All I need is 12 mLs and some fortunate donor. So that's the hard part, but it's not difficult otherwise. So why are you telling me about spit?
Well, actually, Mansi, saliva is much more similar to blood than commonly thought. It actually contains the same components from cells to even the serum proteins themselves. It contains DNA, RNA, proteins, bacteria and viruses, and even cells including white blood cells, buccal cells, and epithelial cells.
That's really interesting. I mean, I hate giving blood personally. I hate blood draws. I have small veins, and I get pricked from the top of my arms right down to my fingers, black and blue afterwards, and in the summer at 110 degrees, I'm wearing full sleeves. So if you can tell me I can move away from this, I'd be very happy.
Well, absolutely. And there's actually more advantages to using saliva than just not getting pricked by needles. Number one, you don't have to go see that doctor or nurse. You can do it in your house at your own convenience. And moreover, the preservation capacity of saliva is much greater than that of blood. Blood, you have to store it at 4 degrees. You have 48 hours to do your experiment. And if you don't, you're going to have to go do it again. With saliva, it's actually stable for months at varying temperatures. And, you don't have to store it and you don't have to worry about it going bad.
Well, blood draws are definitely annoying, and if you can make things this convenient, I can definitely see the advantage of using saliva. But, there's no real alternative for what's really done with blood. You can do a lot of genetic testing with saliva. I think we all know that. There is some HIV testing that can be done with saliva. There's some drug testing. But truthfully, and I have personal experience using saliva for genetic testing, and quite frankly, it hasn't been as successful as it was promised. I've left samples on the shelf for a few months and ended up with a tube full of bacteria. So are you really telling me that that's not the case and that we can actually do more medically relevant things with saliva?
Well, you're absolutely right. Today, with using the current technology for saliva, it's very limited. On the medical side of things, it's really limited to genetics and drug abuse testing for those parents who think their kids are up to no good. When you're looking at research, it's limited to the same type of things. You could do some exosome studies and you do some genetic studies, but that's really all you could do with current technology. If you were able to actually harness the true power of saliva and take advantage of all the components of saliva, you could do so much more. On the medical side, you can do just about everything you do with blood, except the things you do with red blood cells, of course. And on the research side, it's even more interesting because you could do epigenetic studies, you can do sociological studies, you can do exosomes, you can look at biomarker discovery, that you can do pretty much anything that you were able to do with blood before.
Steve, you're blowing my mind here. So spit really does matter, and that's great. Honestly, why would anybody want to have to go to a hospital, get pricked, draw blood, have the hassle of going back for repeat studies? I'm just thinking about my own research. If I can move away from blood draws, I could increase the number of subjects. Subject recruitment would be easier. You wouldn't have to pay them as much either to get this stuff done. So this is great, sounds wonderful, but let's be honest, it sounds a bit too futuristic. Maybe my grandchildren will get to use technology like this.
Well, what makes me really happy, Mansi, is I'm here today to tell you this technology finally does exist. We at Abigen developed technology to collect, preserve, and separate all the components of saliva, from the DNA, RNA, and proteins, to the bacteria, to the viruses, and even to the different types of cells. We can separate out T cells if you want, or if you want a whole lymphocyte population, you can do that too. Furthermore, unlike, as Mansi mentioned earlier, there's no bacterial growth. There's absolutely zero growth due to the components of our preservation solution.
Okay, that sounds great, but tell me a bit how this works. I've seen some of the other saliva kits out there, and there's a lot of steps on how to use them. You're switching caps, you're shaking, you're turning something upside down, you're not turning something upside down. How does this work?
Well, so we developed a sample collection kit with really the user in mind, and ergonomics as well, because a lot of people who are donating saliva have issues with dexterity, quite frankly. This kit is as simple as it possibly could be. You spit in our tube, you close the cap, you shake it, and you stick it in the mail. That's all you have to do. And we tested this kit against other kits on the market today, and as far as usability, ours was always rated higher than the others. Furthermore, we've developed technology to preserve all the components of saliva. So instead of weeks in days, you can actually preserve saliva for months. Again, with blood, 2 days, you have no material left. You can't do your experiment. With our technology, there's really no difference at room temperature when you go even to 3 months out. And in fact, when you do experiments at different times, such as times and temperatures, such as up to 40 degrees Celsius and as low as minus 20, you see really no difference in the yield of T cells, which is what these experiments are showing. Furthermore, we've developed methods to separate out all the components. Think of it kind of like blood processing, where the first steps when you get blood is to, of course, separate out the serum and everything else. We provide methods to do pretty much the same thing, so you can separate out the extracellular material, much like serum proteins, and then you can also separate out the different types of cells for cellular studies.
Okay, sounds great so far, but this is a room full of scientists, so let me reference Jerry Maguire. Show me the data.
Mancy, I'll do just that. So, my first example I'll give you is an epigenetic study we did where we really wanted to see if epigenetically your blood cells from your blood and your blood cells from saliva are the same. So we took individuals and took out their blood and they donated their saliva. We isolated T cells from both their blood and saliva and we compared them at a whole genome level using methylated immunoprecipitation experiments. And what we found is when you look at saliva T cells compared to blood T cells, their epigenomes of all the human known regulatory regions have a have a correlation of about 1. When you take whole saliva, you get that correlation only about to 0.5, something that wouldn't be scientifically usable. Furthermore, we then did qPCR of these different regulatory regions using different probes, and we came up with the same results for blood and saliva, once again showing that a blood cell from your bloodstream and a blood cell from your saliva are the same cell. Just to give you another example, Mancy, is the isolation of white blood cells, kind of our bread and butter at Abogen. We're able to isolate almost 500,000 lymphocytes per mil of saliva and about 16,000 T cells per mil of saliva. We can also do B cells and natural killer cells as well. And these cells are viable for many months at room temperature, and this is before they're isolated out of our solution. And we've done this in over 300 individuals with 100% success. Now, if that wasn't enough, Nancy, I have some more examples for you. So we then did gene expression arrays, and this was kind of to show again that a blood cell from blood and a blood cell from saliva are the same. So once again, we took blood and saliva from same individuals, extracted their T cells, and then extracted RNA and ran array experiments. And what we found is, of course, you get less RNA out of a mil of saliva, of the T cells from a mil of saliva, than of blood, but it's more than enough material to use. But what's great is that the correlation coefficient of, again, blood from cells from blood and blood cells from saliva is pretty much near 1. It's about 0.98 in all subjects tested. So if that's not enough, Mancy, I have some more for you. So I really like this experiment on proteins. So one thing that hasn't been able to be done with saliva before is the use of extracellular proteins. The proteins are very similar to blood serum proteins. So what we did here is we took individuals and we took saliva and we flash froze it, which is the only way you can use saliva and get proteins today. We took the same individual saliva and looked at normal DNA kits that are on the market today, so those that only preserve DNA, and then we used our new kit. And we isolated the different fractions of this kit from the white blood cells shown here to the epithelial cells and bacteria shown here to the extracellular material shown here. And what's great is when you use the commercial kits on the market today, in this lane here, you see that there's essentially no proteins left, you get no banding, and it's completely unusable. When you take the extracellular material using our new kit, you get banding showing that there are in fact preserved extracellular proteins. And you get the same when you look at the white blood cells. So those would be cellular proteins, and these would be cellular and bacterial proteins. And you compare this to a flash frozen sample, which of course is considered 100% when you are looking at saliva. So we are just at commercial launch today, and we have had one researcher use our kit. She wanted to examine the environmental effects of temperature on sports physiology. And this is applicable to things such as NASCAR, where the temperature gets very high in a cockpit. So what she did is she made her subjects exercise. She took blood and saliva, the saliva using our technology, before exercise, after exercise, and during a recovery period. She then looked at the whole lymphocyte counts and looked at the changes. And shown here is her previously published results in blood showing exercise and recovery and the change in lymphocytes compared to baseline. When she did the same exact experiment with saliva, she got the same exact results. And in her own words, she was extremely happy about our technology because it was less restrictive on subject recruitment and caused a less of a burden on her research team and allowed her to do more research overall. Currently today, we're selling two different types of kits. We're selling our S3A collection kit, which is what I've been talking about here, which really preserves all the components of saliva, and we also provide the methods to separate those components. We also sell the S3G collection kit, which is similar to that on the market today and preserves just the DNA. Well, so thank you very much, and I hope after this presentation you can understand why at Abogen we like to say goodbye blood and hello saliva. And please come over and join us if you have any questions after. We're right there. Thank you very much.